in 6 weeks. Now, the defense we've got against infection, of course, the defense is the immune response. The word immunity comes from the Latin immunis, which refers to soldiers in the Roman state who, after they came back from the wars, were actually exempt from tax for a while. They were called the genio immunium. And the tax, of course, that immunity seeks to defeat is the tax of infection. Obviously, microorganisms, viruses, bacteria, and so forth, can replicate and mutate and evolve at a much, much faster rate than we could ever do. So we have to have a very sophisticated, complex immune system to deal with all sorts of possible unknown invaders. And that's basically our immune system. So it divides into 2 bits: the innate immune system, which is rather broad and really reacts against classes of response. And it's very primitive in evolutionary terms. The toll-like receptors, for instance, that are important for NK cells, were discovered in Drosophila, the fruit fly. They were called toll-like because the investigator was Jenny Nusslein-Volhard, thought they were real cool, and evidently toll means something like cool in German. There's a lot more interest in adaptive immunity than there was, say, 20, 30 years ago. In innate immunity, rather. The reason for that is that as we've become much more sophisticated in our understanding, and we've got much better probes, and we've been able to look for the cytokines and the chemokines and all those things. You know, when I started in immunology, the only cytokine was called macrophage inhibitory factor, MIF. In fact, John David, who discovers it, says it should be called IL-0, but they never did that. I mean, and then there was IL-2, and that's all there was. We knew about the type 1 interferons from the work that went on early with Isaacs and Lindemann and so forth, but we didn't know gamma interferon. A lot of stuff we didn't know. And then, of course, the cytokines, the chemokines. Science was so much fun back then because we didn't know anything, and you could speculate, and you could have a lot of fun. Now you actually have to do the work. Adaptive immunity, much younger in the evolutionary sense, and until recently, we were way ahead with our understanding of adaptive immunity, way ahead of innate immunity. And of course, now, as you realise, a lot of the focus in immunity is to try and understand how what goes on during that innate immune response sets up the adaptive response. And the reason we really want to understand that, apart from understanding the disease processes, is we want to make better vaccines. And if we can understand the really important factors that are going on in that innate phase, that really non-specific phase when the viruses are infecting the cells, all sorts of stuff being produced and so forth, if we can understand that better, maybe we can make better vaccines in the longer term. The other reason we want to understand the innate response, particularly in influenza, is that cytokines themselves and chemokines can contribute to pathology. And then, of course, there's a whole new world of biology that's come in with the discovery of, via interleukin-1, the discovery of what we call the inflammasome and all that stuff. So it's a pretty fascinating and very increasingly complex time. And of course, we all get increasingly specialised as we go on with this. The vaccination, the basis of vaccination, of course, is immune memory. We have, in the lab mouse, for instance, we have lifelong T cell and B cell memory. The T cell memory is in the cells themselves. They're recirculating all the time. They're present at a higher frequency than they would be in a naive, previously unexposed animal. B cell memory is in the B cell, the plasmablasts and plasma cells. The plasmablasts or the B cell memory cells sit in the lymphoid tissue, they recirculate in blood. The plasma cells, in the very long term, after the antigen is out of the system, the virus is out of the system, the plasma cells will tend to sit around the bone marrow and pump out antibody from there. And we know from humans, people have gone back and looked, particularly Raphael Med's group in Emory, you can find that people who were exposed to, say, yellow fever virus or vaccinia virus 50 years previously still have memory T cells and are still making antibody against those pathogens. We know that when the H1N1 flu viruses came back in 1970-something it was, that people who'd gone through the 1918 epidemic still had antibody. And so this memory is very, very long-term indeed. It works by protein-protein interactions, the typical antibody molecule that binds to, say, the surface glycoprotein on a virus. They tend to bind to tertiary structure. That's the Coleman-Laver group and Coleman's group and Labor's structure of antibody binding to the neuraminidase protein that led to the development of the drug Relenza. On the other hand, the T-cell responses that I study, we think of them as recognising more linear epitopes. That actually is a misconception. What they're actually recognising is also tertiary, but it's the peptide bound to another protein that's the actual structure. Where we stand with vaccines, we've got good vaccines against viruses that don't vary very much and cause systemic infections. So we've got a great vaccine against polio, for instance, because the virus doesn't change very much, gets in normally, infects us in the pharynx and gut and respiratory and gastrointestinal tract, only causes disease if the virus can go through the blood, get to the central nervous system, destroy nerve cells. If we've got antibody in the blood due to the vaccine, it doesn't get there, we don't get polio, we're fine. Polio vaccine is very successful. Measles vaccine is the same. The virus normally comes in through the oropharynx. It has to get round in the blood to get to the skin, to get to the brain, to get to the various places that cause problems. You've got antibody in the blood, it's fine. Yellow fever, the same. Viruses injected by a mosquito. Where we get into real trouble, we have more difficulty with viruses that grow only on mucosal surfaces like flu, but we can make vaccines that work quite effectively as long as they're specific enough. And even with human papillomavirus, which is a virus which grows in desquamating epithelium in, say, the female reproductive tract, we've been able to make a good vaccine, or Ian Fraser and his colleagues have been able to make a good vaccine. Where we're in real difficulty is with viruses, particularly systemic viruses, that change within the individual and have a capacity for persistence. And the two that we think about particularly are human immunodeficiency virus and Hep C virus. And these are enormously difficult challenges for immunology. In fact, whether immunologists in the end are going to do anything really useful with them is a moot point. We know we can deal with HIV with drugs. We haven't done that well with HCV, I guess, but I don't know where actually designer drug therapy sits for that virus. Now, what I work on, I've worked on over the years, is the virus-specific cellular immune response, the killer T-cell response. These are the white blood cells that go around in the blood, they go into tissues, and they destroy virus-infected cells. Now, we have to destroy virus-infected cells. Viruses, as you know, are obligate intracellular parasites. They can only grow within living cells, and each virus-infected cell is... pushing the wrong buttons. Each virus-infected cell is actually a factory. So one virus particle can go into a cell, 1 million can come out. You've got to destroy that factory. And the way that the factory is destroyed is by the specific T-cell receptor, which recognises the change on the surface of the cell. The T-cell tells the virus-infected cell to commit suicide. It triggers the cell death pathway. It does that via molecules called perforin, which is a channel-forming protein, and by other molecules, particularly serine esterases, which go under the name of granzymes. These go across into the target cell and trigger that caspase pathway that actually leads to the classical apoptotic nucleus. So it's not really a killer T-cell, it's an inducer of death, an inducer of suicide T-cells. So a smart lawyer can make something of that, I think. But it's best not to focus on smart lawyers. These are T-cells bouncing around on some tumour cells on a dish. This is a time-lapse photo by Misty Jenkins. And here you can see a killer cell going to kill that. And now that's over a matter of hours. It can vary a lot between the actual conjugation of the two cells until you actually get the death event. It could be quick, it can be long, it will vary for different antigens, and it's really, I'll play that again, it's the best part of the talk. There we go. Yes, that's right. Nothing like a bit of death. I mean, I find out, as I've got older, the only books I can read have to have a murder in them. That's right. It says something about the human condition, doesn't it? So, of course, if you've got cells that are going around killing other cells, you've got to be very careful to target them precisely. You don't want promiscuous killing. You don't want to kill a cell running around and being a mass murderer. And so you want it targeted to that cell that you need to take out. And the way that that works is basically the T cell, and I guess everyone knows this now, but of course this basically gets back to what we discovered that won us the Nobel Prize, that the T cell focuses on peptide from the viruses presented in the tip of the major transplantation antigens or the major histocompatibility complex glycoproteins. These, of course, are the molecules on our surface. We have each of us has 3 loci, the codes for these molecules, the class 1 MHC molecules or strong transplantation antigens. As we have 3 loci, and most of us are not highly inbred, well, none of us are. I mean, we're F1s. We're not mice. We don't do back crossing. And the Egyptians did that for a while, I believe. But well, brother-sister matings anyway. But we're F1s, which means we've got 6 of these things. We've got 6. Chickens are interesting. They've only got 1 locus and 2 glycoproteins. And you can actually see very clearly in chickens that there is a very clear association between disease susceptibility and MHC type in chickens. It's much, much harder to see in humans because we've got 6 possibilities for giving us immune response. But here we see the T-cell peptide MHC interaction. This was published actually the year that Zinkenagel and I got the Nobel Prize in 1996. It came from Ian Wilson in California. Here we've got the T-cell receptor, here the MHC molecule and the peptide. And of course, these molecules, the MHC molecules, not only do we have multiple loci, 6 loci, we also have multiple alleles. They're extremely polymorphic. And so there's an enormous number of these genes. With the chicken, there's still a lot of polymorphism, even though they only have 1 of these rather than 6. But what also changes in the chicken, which doesn't change in us, is a molecule called tabazin, which is involved in loading that peptide onto the MHC. And because they've only got 1 gene and 1 locus, the tabazin molecule can co-evolve with it. If you've got 6 different or 3 different loci, then your tabazin molecule has to be like a sort of general servant, if you like. You know, the plumber. It has to fit 3 different or 6 different class 1s. So it has to be able to work with them. But if you've got a single one, it's like a personal valet, and it can sort of evolve with them. So that's one of the ways that this co-evolves. The chicken studies are actually wonderful. They're done by a guy called Jim Kaufman, who is now at Cambridge, who's a terrific guy. And if you ever know of Jim coming out here, if you really want to get a fascinating talk by a very interesting character, he's just been out recently. Did he come down to Geelong? He should have. He spoke in Melbourne. Jim is just fantastic. Also a wonderfully nice guy, actually. This is, back in the primitive days, this is how we saw T-cell recognition. This is before, I just should say this for the young people. But this is 1974, this was published. That's a T-cell receptor and that's the MHC thing. We had no monoclonal antibodies. We had no recombinant DNA technology. We had no sequence for the MHC glycoproteins. We had sequences of things like myoglobin and milk proteins where you could get buckets of the stuff. But if you don't have recombinant DNA technology and you don't have monoclonal antibodies and you want to do the sequence of a cell surface glycoprotein, you've got a real problem because the machinery was also much more primitive and we didn't have the computing and all the rest of it. And I think the first sequences of MHC came along about 1978 or 79, and we were delighted when we got 23 amino acids, if you can imagine. And so it was very primitive days. We made some good guesses, actually, and we were lucky. And we're also very lucky because there's all these differences in the transplantation molecules, we were able to tie it into the transplant system or the specific immune recognition because the transplanters, the people studying transplantation, had developed a wonderful spectrum of recombinant mouse strains, inbred and recombinant mouse strains, to study graft rejection. It was really started by this guy, George Snell. George went to the Jackson labs in Bar Harbor, Maine, the mega mouse labs, if you like, in the 1930s, and he wanted to study immunity to cancer. So what he was doing, he was inducing cancer in mice by painting them with methylclanthrin, which gives you a carcinoma, a whatever it is, well, some sort of damn tumour. And then he was transplanting that tumour, and he saw very rapid rejection. And he knew this couldn't be tumour immunity because tumours are never rejected like that. So he worked out it was the difference between the mice. And he thought he'd take a few months off and work out what that was. And of course, he was still working on it when he retired at age 80 or whatever. And they developed all these mouse strains, which we were then able to get, which isolated each of those genetic loci and the genes. And so we were able to, when we got an effect which was mapping to the MHC in a broad sort of way, we were able to get all those recombinant and genetic mouse strains, just infect them with virus and map it very quickly to the class 1 genes. So We actually came along and told the transplant people what the MHC was for. They